Hello, I'm Nadia, Akadeum's Associate Director of Development Chemistry. Today we're taking a closer look at an integration leveraging the cell processing of the Cytiva Sepax C-Pro and the gentle cell isolation of Akadeum Human T Leukopak Isolation Kit GMP. This integration was specifically designed to streamline your cell therapy workflows, maximizing cleanroom footprint and ensuring workflow flexibility while maintaining regulatory compliance and ultimately yielding better therapeutic cells. What makes this workflow a significant improvement over your current process? It all comes down to the automated closed system cell washing with the Sepax and Akadeum's unique buoyancy-based microbubble technology, all with GMP quality reagents and a clinically compatible workflow. The Sepax system offers precise fluid handling, reduced manual intervention, and consistent cell processing, which are crucial for maintaining cell viability and reproducibility in GMP environments. Akadeum's microbubbles gently bind to unwanted cells, floating them to the top of the processing bag. This combined approach offers fewer steps, minimal cell handling time, and a truly scalable cell isolation and washing process with untouched T cells that integrates directly into your existing closed system GMP-compliant workflows. Inside the Akadeum Human T Cell Leukopak Isolation Kit GMP, you'll find our strap-evident microbubbles, our negative selection antibody cocktail, and buffer. For this particular workflow, you'll also need standard cell processing bags, an end-over-end rotator, a tube welder, and of course the Cytiva Sepax C-Pro system and associated kits, none of which are included in the kit. That's how simple it is to integrate the Akadeum T-Cell Leukopak Isolation Kit GMP grade with the Cytiva Sepax C-Pro Cell Processing System. The Cytiva Sepax C-Pro Cell Processing System seamlessly initiates a GMP automated cell processing followed by Akadeum's innovative microbubble technology for efficient 16-minute isolation and automated closed system washing. This combined approach provides efficient, high-quality T-cell isolation and maintains the health of your starting sample with isolated T-cells ready for your downstream applications, whether that's cell culture, expansion, gene editing, or other functional studies. To learn more or request a demo, visit us at akadeum.com.